The trip up to Christmas Island was actually kept secret from the crew, although the officers and the radio operators had prior knowledge of that. So there was quite a lot of speculation as to where we were going. Not far out of Auckland, probably the second or third day, we were told where we were going and what the mission was, and almost immediately we started exercises to close down the ship, get the crew into what they called shelter stations, which were compartments deep within the ship. All circulation of air was cut off to stop any contaminants entering the ship. And the idea was also to get men who would be on deck at the time of the detonation to their shelter stations as quickly as possible. And so they were continually trying to reduce the amount of time that it would take the crew to get to their shelter stations and only be exposed to a minimum or no radiation. This continued right up to the period that we arrived at Christmas Island, and even during the carrying out of duties at sea before the first detonation and in between every detonation as well. The first detonation was something of a rather tense affair. The crew, except the very few that were required to keep the ship running, were lined up at what they called blast stations, which were predetermined positions on the port side of the rail of the ship. And we sat there with our backs to the area where the detonation would take place. We were dressed in what was then described as protective clothing, no skin showing, and dark goggles. Close to the period, close to the time that the bomb was to be dropped, the bomber pilot was connected to the ship's intercom, and he gave a running commentary as he was approaching the position. He told us when the bomb had gone. There was a countdown, and right on the spot of 0, there was this horrific flash, and by that time we'd been, as we'd been trained to do, we also had we were sitting in a semi-fetal position and had our hands over our eyes. That flash was so bright that you actually saw the bones of your hands through the dark goggles and your hands, which came as a bit of a shock to a lot of people, including myself. After a few seconds, we were then told to stand up and turn around and face the blast, which we did. And what we saw was a round cauldron of fire, really, rising up into the sky, and as that progressed, the normal spiral that forms below it and gives it its mushroom shape was forming as well. I sort of remember at the time, just as a reaction, looking around me, probably in a useless effort to see if there was anything to be seen, probably radiation, but of course there wasn't, and even if there was, you wouldn't feel it anyhow. So really, that was the first experience, and as time drifted on this very carefully selected day for calm weather, etc., the clouds slowly dispersed, and we made our way towards it, actually, and passed through Ground Zero. And on the other side, waiting, was HMS Warrior, the aircraft carrier and flagship of the task force. And by that time, we'd packed up all monitoring equipment, and that was passed over to the Warrior for analysis. It became obvious after the second blast that we were in fact being used as part of the experiment. The ships were required to go through Ground Zero. Later research detected the passages of the ships had also sailed under the projected radiation fallout path. And as there were 9 tests in all, and after each test, the ships were progressively moved closer to Ground Zero, the point of detonation, and the protective clothing was reduced, so that by the time I witnessed the last one that I was going to do before I left the ship, Grapple Y, the 5th detonation, I observed it dressed only in shorts and sandals. The final detonation, the 9th one, the ships were only 20 miles from the point of detonation.
One of the dangers of exposure to radiation is harm to the chromosomes. The chromosomes break, and that's not good. And translocations is the term given to where pieces of chromosome break off and go across to another chromosome. And most cancers come about as a consequence of chromosome translocations. And the best technique for detecting the occurrence of translocations on chromosomes is using this technique called MFISH, multicolour fluorescent in situ hybridization. And here's how it works. What we do is we buy DNA from commercial companies who have extracted the DNA of each of the chromosomes specifically, and they have added a coloured dye to each of the chromosome 1, the DNA of chromosome 2, that has a different dye attached to it, chromosome 3, a different dye. So we have all of these, the DNA separately for chromosome 1, 2 and 3, each with a coloured dye added to it. That's what we call our chromosome paint. Now the beauty of DNA is that if you heat it, the DNA helix will part, and under the right conditions you can flood the DNA, flood the chromosomes on your slide that you've prepared in advance. You can flood the slide with a cocktail of your paints. And those paints, the DNA, under the right conditions, will hybridize to the chromosomes on the slide that you've prepared of the veteran or control or whatever person you're studying. And then you can prepare the slide, make it permanent, and look at it under a fluorescent microscope. The paints fluoresce all these different colours. And so when you look at the chromosomes under a fluorescent microscope, you can see all the different colours. Chromosome 1 is yellow, and so on and so forth. And if there has been a translocation, it is simply a matter of looking to see a switch in colour from one chromosome to another. And it is called in situ hybridization because the hybridization is occurring at the site, in situ, at the site of where it occurs on the chromosome on the slide. And it's fluorescent because the paint fluoresces hybridization is because you've hybridized the DNA of the paints to the DNA of the chromosomes on the slide. What it involved was collecting the blood from the veterans. So we travelled all around the country, New Zealand, collecting small blood samples of each of the veterans, like a couple of teaspoons of blood, and in sterile tubes. And they were brought back to Palmerston North, to Massey University. And they were collected independently, and the blood was coded. The tube was coded separately by the Massey University Health Centre, so that we did not know if a sample was from a control or from a veteran. So this is called a blind study, and this was very important to protect ourselves from accusations of bias. So we did this for the whole study. We did not know, when we were looking at a preparation, whether it was from a veteran or a control. And it was not until the end of the study that we opened the books, so to speak, and then analysed the data. So it's very important when you conduct a study like this that it must be conducted blind. You must not know whether the subject is a veteran or a control. It's also very important when you're designing the study that you select very good matched controls. And that's a very important word, matched controls. Because what you're trying to do is to isolate the variable of interest, which in this case was this group witnessed the bomb test and this group did not. But you must isolate that variable by eliminating any other confounding factor which may play a role in interpreting your results. For example, age is very important. So the men had to be of very close age, similarity in age. Where they live is important. So we selected a control from the same town or city where a veteran lived, because it might be something, there may be something in that town which is causing chromosome damage, so you have to take that into account, and that's what we call matched controls. So these are factors which are very important in designing your study. So not only that, there was cigarette smoking, alcohol consumption, exposure to organic solvents. Perhaps a person may have been a painter. So if the veteran became a painter later on in life, then we had to also have a control who was a painter. So that if anyone came along and said, "Oh, your results, you can't trust them because the veterans were heavy smokers," we said, "All right, let's look at the matched controls." And the matched controls they smoked to the same degree. So if we found a difference in translocation frequency, we know that it cannot be due to cigarette smoking because both groups smoked the same amount. So this took us about a year to interview all the prospective participants and isolate all these variables and plan it meticulously. And I'm very grateful to my colleague, Professor John Pott, who has a wealth of experience in this area of studying human populations. He planned it meticulously, and that has been just as important in the success of the study as is the science of doing EM fish. When we collected the blood samples from the participants, we then cultured the lymphocytes. So we took a drop of blood and under sterile conditions, put a couple of drops of blood into a tube, sterile tube, with a culture medium, with a stimulant to stimulate the blood lymphocytes to enter into cell division. And we left the culture at 37 degrees in an incubator for 3 days. And after 3 days, we then took the tube and added some colchicine, which arrests the cells at metaphase. We left them in the colchicine in there for 1 hour, and then we harvested the chromosomes. We took the tube and we centrifuged the lymphocytes down, and then using a pipette we drop the lymphocytes onto a slide and let it dry, and that is how we prepared the chromosome. Yes, we had a special software program that cost $30,000, very expensive, which photographs your spread of chromosomes, and then it will pair the chromosomes up on the screen for you to construct what we call a karyogram, although we do use the word karyotype as well. And you can see the pairs of chromosomes, each with their different their colours, and you can see if any translocation has occurred. And the software program we have can isolate a chromosome with a translocation and analyze the fluorescence of that chromosome and tell you specifically which chromosome the fragments making up that chromosome come from. It might be a translocation between chromosome 1 and chromosome 14. It might be a combination of yellow and blue. So it's quite powerful.
They used to do drills where they would hide pellets on the boat, and this man had to go around with a Geiger counter to see if they could find where the pellets were, just so that they could practice cleaning the boat properly. This is an example of M fish, and what you do is you take a blood sample, set up a blood culture, a few drops of blood with growth stimulants, that PHA, phytohemagglutinin, to stimulate the lymphocytes into division. You let them grow for 3 days, then you add colchicine, and that halts the metaphase, and then you drop them on a slide. Then you, 37 degrees, you add your cocktail of the DNA, the individual chromosome DNA with the dyes added. Then you heat it to 58 degrees and you hold it there for about an hour, and then you lower the temperature and hybridize. Anyway, that's just technical details. When you look under the fluorescent microscope, you see all these chromosomes. The software package we have will take that photograph and cut out the chromosomes and pair them up. So that's got a yellow dye added to it, that's a green, and these are individual dyes, those 5, and then you've got a combination of green and yellow to give a slightly different one fluorescent spectrum to that one, and this is 2 dyes added onto the DNA, and these are 3 dyes. Occasionally you will find a preparation like this. If you look at this chromosome here, it's made up of a yellow chromosome and a part of a blue chromosome. So our software program cuts them out and pairs them up, and what we find is here's the one chromosome with a bit of the blue on it, and that is of chromosome 14. Chromosome 14 has the centromere near the end, and what has happened is that the chromosome 14 has broken next to the centromere, and the blue part of the other chromosome 14 has gone on to the end of this chromosome 1, which is broken here, and the centromere of this chromosome has gone on to the end of the yellow bit that's broken off from here. Now this looks yellow, which is confusing, but just ignore it, because its centromeric DNA is made up of a special kind of non-genetical DNA, which can give all sorts of funny colours. But anyway, so this is what's happened here. Then you can take that chromosome, isolate it on your computer, and the software package we did will analyze that chromosome, and it will tell you that this part is chromosome 1, and this part is from chromosome 14. And it tells you the different 3 dyes that are making up that blue, and the 1 dye that's making up the yellow. This is confirmation that that's chromosome 14 and that's chromosome 1. And this is what the chromosome looks like under the phase contrast microscope.
The program actually cut all these.
Yes, cut them all.
You don't have to do it yourself.
This chromosome here, there's been a deletion. See in chromosome 13, that's short. There's a one-way translocation. There's been a break next to the centromere here of chromosome 2, and it's ended up on the end of that. Oh, there's the other one of 3 lying over there. But when you look at this chromosome here, and you do your analysis of it, it tells you what dyes, the 2 dyes making up the brown, and identifies it as chromosome 8. The brown is chromosome 8. This identifies chromosome. And if you went to, it tells you it's chromosome 8. It's hard to read, and it's chromosome 2. So that's a one-way translocation.
What is this?
That's the fluorescent spectrum that the software program has scanned the chromosome and looked to see what combination of what fluorescent spectrum is making up that chromosome.
And the black and white over there?
That's the normal chromosome. When you remove the dye, that's what it would look like. What's called the Giemsa banding pattern of these two chromosomes, but it's not a reading. No, it's not a reading. Yeah, this is a real image of the chromosome without the dye. If you have an ambiguous result, you could be subjected to criticism. So they may say, "Oh, you're just making that up. You don't know for sure that it's those two chromosomes involved." So, in which case we played safe and just eliminated that from our data. So you might say that our results are conservative. The actual data is worse than what it looks. Our view is that for such a high amount of translocation to occur, we believe that there must still be radiation in the blood of in the body of the veterans, probably in their bone marrow.
But isn't that something you can measure?
You could, and they should do. But the thing is with radiation that if you did that test, the radiation is so powerful you may not pick it up. It may be there, but you may not detect it in the body. So the veterans are reluctant to have that test done because it may be that the results may show they have no radiation in their body, and in actual fact they do. So that could be the case. I'm not saying it is, but that could be. If they didn't have radiation in their body today, then the only explanation for this is that these are long-lived lymphocytes which we have stimulated into division, which have remained dormant for 50 years. And some lymphocytes have been known to live for that long, as they are found in Japanese studies with the Hiroshima survivors. But I think it is more likely that these are new cells which have suffered radiation exposure because there's so many of them.
The cells are not stem cells,
No.
It's normal cells.
Just normal lymphocytes, yeah. They're just normal lymphocytes.
But it's not stem cells
No.
you activate.
No, these are normal lymphocytes which are maturing and
because
new.
You wouldn't find the stem cells in peripheral blood.
No.
So therefore you can say it's not stem cells.
That's right, they're not stem cells.
But if you had not activated it for division, you wouldn't be able to see anything. Of course.
You wouldn't. The only way you could see the translocations is to stimulate them into cell division.
Could it be done with some other type of cell?
Yes, skin cell.
Skin
Good
cells.
question. But the most reliable method is blood. But I don't know if the skin cells would show these results because skin is replenished so quickly. It is better to do blood. It's easier to do blood anyway. You can take samples quite quickly. So what is this one? One-way translocation between 21 and 22. The smallest chromosome in the genome is chromosome 21, and 22 is slightly bigger. And this is from a mistake that was made in the early classification of human chromosomes. They numbered them 1, 2, 3, etc., according to the total length, with number 1 being the longest, 2 being the next longest, and so on, decreasing in length. And they made a mistake when they got to these two, and they thought that 21 was longer than 22. But in actual fact, careful studies have since shown that 21 is shorter than 22. There's the X chromosome and there's the Y. And of course all these men are XY. Many studies using just the three of chromosomes And there have been lots of studies done on Japanese survivors from Hiroshima, Chernobyl cleanup workers. There have been studies done in Germany, but this is the first time where there's been a defined population of people on the boat. Well, we know where they were, and they were witnessing an atomic bomb explosion.